video, we are going to learn about some qualitative analysis tests, some tests related to carbohydrates in particular. So let's start. Reactions of monosaccharides. Carbohydrates are hydrophilic substances with potential or free aldehyde or keto group and a number of hydroxyl groups. Monosaccharides, the basic component in the series, have a single carbon chain glucose and aldohexose and fructose, ketohexose. All the most common monosaccharides perform the test with solution of glucose and fructose. So Molish test. This is general test for all carbohydrates. Carbohydrates, when treated with concentrated H2SO4, undergo degradation. This is Molish test. This is the most basic test or general test for all carbohydrates. Carbohydrates, when treated with concentrated H2SO4, undergo degradation to give a furfural derivative. Hexose, for example, form dihydroxymethyl furfural. These compounds condense with alpha-naphthol to form a colored product. To 1 ml of glucose solution, add 2 to 3 drops of 1% alpha-naphthol. Mix and add it carefully through the sides of the test tube. 2 ml of concentrated H2SO4 is added so as to form 2 layers, not the formation of a reddish violet ring at the junction of the 2 liquids. Repeat the test with fructose. Here could say structure of hydroxymethyl furfural, alpha-naphthol treatment. Chromogen is formed. Chromogen in the sense colored compound. Next major test is Benedict test. Benedict's. Reducing sugars under alkaline condition tautomerize and form enediols. The enediols are unstable and decompose to yield a variety of products. 1,2 enediols will give formaldehyde and pentose. The chain reaction continues to produce shorter chain aldehyde, which are powerful reducing agents. They can reduce the cupric ions to cuprous ions, which is the basis of this Benedict test. The cuprous hydroxide formed is not easily soluble. So, in order to keep the hydroxide ion in solution, a metal chelate like citrate tartrate is introduced into the solution. So, Benedict solution contains the milder alkali Na2CO3. When reducing sugar solution is treated with Benedict's reagent, the sugar is decomposed and the product reduces cupric ions to cuprous ions as yellow cuprous hydroxide. Then sugar enediol Cu2 plus CuSO4 oxidized state to form sugar acid in an enediol. Then reduced to form Cu2CuOH, and that will turn to Cu3. Okay. So cuprous hydroxide during the process of heating is converted into red cuprous oxide. The Benedict's reagent test is more specific than Felling's test. To 2 ml of glucose solution, add 3 ml of Benedict's reagent. Mix and boil 4 minutes over a small flame. Note the formation of red precipitate. Repeat with fructose. In Buffoyt's test, aldoses and ketoses can reduce cupric ions even in acidic conditions. Monosaccharides react very fast, where the reaction with reducing monosaccharides is low. This property is used in Buffoyt's test to distinguish reducing monosaccharide. Once again, I am going to read. Aldoses and ketoses can reduce cupric ion even in acidic conditions. Monosaccharides react very fast, whereas the reaction with reducing monosaccharides is low. This property is used in Buffoyt's test to distinguish a reducing monosaccharide from a reducing disaccharide. Monosaccharides react fast. So Cu2 plus formed is treated with phosphomolybdic acid. The colorless phosphomolybdic acid is reduced to blue phosphomolybdic acid. Perform a test simultaneously with glucose and fructose 2 ml each of the glucose and fructose solution is taken in test tube and add 2 ml of a Baffoit's reagent to each. Keep the tube for 3 minutes in a boiling water bath. Cool under running water. Add 4 molybdic acid drop by drop with mixing till the solution is clear. A deep blue color is formed in both the instances. Deep blue color. Okay. Then Selivanov's test. Next one. Selivanov's test. Composition of Selivanov's reagent. Resorcinol in dilute HCl. HCl and Selivanov's reagent dehydrates the ketose to form furfural derivatives which condense with resorcitol to form cherry red color complex. This test is used to distinguish between aldoses and ketosis. Prolonged boiling may also give a positive Selivanov's test for aldose due to their conversion to keto group by HCl. Then ozazone test. That means prolonged boiling should be avoided, I guess. We will be learning about that later. So ozazone test next. Principle: when reducing sugars are treated with phenylhydrazine, first phenylhydrazones soluble are formed. On heating, these hydrazones further react with 2 molecules of phenylhydrazine to form sugar ozazone, which is insoluble. Non-reducing sugars like sucrose does not form ozazone. Sucrose does not form ozazone. Here you could see a picture which represents the glucose ozazone or fructose ozazone, whatever it may be. We will be specifically drawing this two later. Okay, don't worry about that. Then glucose plus phenylhydrazine, glucose phenylhydrazine, deoxy sugar, phenylhydrazine, and again treating with that. Then glucosazone. For distinguishing the different reducing sugars in urine under the condition of glycosuria or lactosuria, lactose increase or glucose increase, this is the only test to differentiate between maltose and lactose. Only reducing sugars which have a free aldehyde or keto group will form ozazone crystals. Glucose, fructose and mannose form identical ozazones. This is because these sugars differ only at carbon numbers 1 and 2. Ozazone formation involves C1 and C2. So the different features these sugars at C1 and C2 are masked during ozazone formation. So glucosazone and fructosazone crystals are formed during boiling itself. So maltosazone and lactosazone crystals are not formed during the process of heating, boiling, because ozazone of these sugars are soluble in hot solution. Hence, ozazones of maltose and lactose are formed only on cooling. So, glucose, fructose and mannose. Just remember this fact. Ozazones of maltose and lactose are formed only on cooling. Okay. Glucose, fructose and mannose give a positive test for ozazone reaction. All of them form colored needle-shaped crystals, hence they are brightly insoluble. These crystals are named glucosazone, fructosazone and mannose respectively. And reactions of glucose. Now we are specifically dealing with the glucose only. Okay. We will be writing up the experiment, later its observation. What is the inference obtained from that? Okay. So first we will be performing a solubility test with this sample of glucose. Most probably a liquid sample or a solution sample, like whatever available glucose powder or whatever it may be, it's dissolved in water and it's provided. So solubility test is performed. Test with solubility of given substance in water. It's soluble. We know that glucose is easily soluble in water. That means absence of polysaccharide, because polysaccharides are insoluble in water. Okay, then we will be performing Molish test. Molish test, we just learned the principle and the importance of Molish test. So Molish test, how to perform this? Take 1 ml of test solution in a clean and dry test tube and add 2 to 3 drops of Molish reagent. We will be introducing the Molish reagent into that. And mix well. Now forget the quantity. 1 ml of test solution in a clean and dry test tube. And after that add 2 to 3 drops of Molish reagent. Okay. Then mix well. And then add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube. Here we are focusing on creating a circular layer. So we won't be mixing the H2SO4 directly into the solution. We won't be shaking that. We will be just introducing this concentrated H2SO4 along the sides of the test tube without shaking. Don't shake. Okay. Once again, take 1 ml of test tube in a clean and dry test tube. Take 1 ml of test solution in a clean and dry test tube and add 2 to 3 drops of Molish reagent. Mix well and add 1 to 2 ml of concentrated H2SO4 along the side of the test tube without shaking. A reddish violet ring at the junction of two liquids. First we added 2 to 3 drops of Molish reagent, then 1 to 2 ml of concentrated H2SO4 along the sides. That will create a reddish violet ring at the junction of two liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condenses with alpha naphthol to give a violet ring at the junction of the two liquids. Alpha naphthol. Here you could see the reaction. Try to draw this if possible. And then colored compound or chromogenous formed. And Felling's test. To 1 ml of test solution, add 1 ml of Felling solution A and B. Mix well. Heat in boiling water. Okay. Reddish brown precipitate formed. Presence of reducing sugars. In alkaline medium, carbohydrates with free aldehyde or ketone group reduces the cupric ion to cuprous ion in cupric hydroxide formed is concentrated to cuprous oxide, which gives reddish brown precipitate on heat. Reddish brown precipitate on heat. Cuprous ion, the cupric hydroxide formed is concentrated to cuprous oxide, which gives reddish brown precipitate on heating. Cu2 plus Cu plus COH Cu2. Then we are going to perform Benedict's test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution. Boil it for exactly 2 minutes. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution. Boil. Brick red precipitate is formed. That indicate presence of monosaccharide. In the presence of an acid medium, the carbohydrate with free aldehyde or ketone groups reduces the cuprous ion in cupric to cupric ion. The cuprous hydroxide thus formed is converted to cuprous oxide, which is red scum on heating. Later, we will be performing Moissan test. Take 3 ml of test solution in a clean dry test tube and add 1 spatula of phenyl hydrochloride on equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Once again, take 3 ml of test solution in a clean dry test tube and add 1 spatula of phenyl hydrochloride, equal amount of sodium acetate and 2 to 3 drops of glacial acetic acid. Mix well and keep the test tube in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tubes to cool under tap water. Take out the crystals with the help of glass rod. Mount on a glass slide using cover slips and observe under the microscope. That will indicate needle or broomstick shaped crystals. Presence of glucose. Glucose reacts with phenyl hydrazine sodium acetate mixture 1:1 ratio to form crystalline glucose zone. Then next is about reactions of fructose. Okay. So first we are going to perform a solubility test with the sample of fructose given. Test the solubility of the given substance in water. Soluble. It's turned out to be soluble. Then next Molish test is performed. That solubility is indicating the absence of polysaccharide. When Molish test is performed, take 1 ml of test solution in a clean dry test tube and add 2 to 3 drops of Molish reagent. Mix well and add 1 to 2 ml of concentrated H2SO4 along the sides of the test tube without shaking. A reddish brown ring at the junction of two liquids. Presence of carbohydrates. They are dehydrated by concentrated H2SO4 to form furfural or hydroxymethyl furfural. This condensed with alpha-naphthol to form violet color at the junction of two liquids. Colored compound or chromogen. And Felling's test. To 1 ml of test solution, add 1 ml of each Felling solution A and B. Mix well and heat on boiling water bath for 3 minutes. Reddish brown precipitate is formed. Presence of reducing sugars. In the presence of alkaline medium, the carbohydrate with free aldehyde or ketone group reduces cupric ion to cuprous ion. The cupric hydroxide formed is converted to cuprous ion, which gives reddish brown precipitate on heating. Then Baffoit's test. Take 1 ml of test solution in a clean and dry test tube. Add 2 ml of Baffoit's reagent. Mix well and boil it for 1 minute. Red scum is formed. So presence of monosaccharide in acidic medium. The carbohydrate with free aldehyde or ketone group reduces the cupric ion to cuprous ion. The cupric hydroxide thus formed is converted to cuprous oxide, which gives red scum on heating. And Celavano's test, a new test, I guess. Take 3 ml of Celavano's reagent in a clean dry test tube and add 1 ml of test solution. Mix and boil for 30 seconds. Cool the test tube and observe. Cherry red color, not violet. Cherry red color. Presence of fructose in acidic medium. Ketosis on reaction with acids give furfural derivatives, which further reacts with resorcinol to form red colored complex. So ketose, acid, or resorcinol colored complex. Then Folger's test. I'm not sure about the pronunciation. To 1 ml of the test solution, add 3 ml of Folger's reagent. Heat in a boiling water bath for 3 minutes and cool. That will create a blue color is obtained. Presence of fructose, ketose on reaction with acid gives furfural derivatives. They condense with Folger's reaction reagent to form a colored complex. You could say a picture which depicts formation of fructose. Then Ozone test. Take 3 ml of the test solution in a clean dry test tube and add 1 spatula of phenyl hydrazine hydrochloride and add 2 to 3 drops of glacial acetic acid. Mix well and keep the test tubes in boiling water bath for 20 minutes. Note the formation of yellow crystals. Allow the test tube to cool under tap water. Take out the crystals with the help of a glass rod. Mount on a glass slide using the cover slip and observe under microscope. You could see yellow thin needle or broomstick shaped crystals. And presence of fructose. Fructose reacts with phenyl hydrazine sodium acetate mixture to form fructose. Fructose 3NH2NHC6H5 create fructose. And next, reactions of disaccharides. Reactions of disaccharides. Disaccharides are formed by the union of two monosaccharide units by glycosidic linkage with elimination of one molecule of water. Glycosidic linkage. It may be reducing or non-reducing depending on the availability of free or potential aldehyde or keto group in their molecular structure. The disaccharides of biologically important ones are maltose, lactose, and sucrose. And a Molish test. Composition of Molish reagent: 1% alpha-naphthol in 95% ethanol. Principal carbohydrates with the concentrated H2SO4 undergo dehydration to give furfural or furfural derivatives. This compounds condense with alpha naphthol to form a reddish violet ring. This is a general test for all carbohydrates. Excess alpha naphthol solution and impurities in the reagent may give a green colored ring. Molish test is given by carbohydrates with at least 5 carbons. Therefore, trioses and tetroses does not answer this test. So, interaction of acid with water produces heat and can cause chaining of carbohydrates, resulting in the formation of a black ring. Therefore, acid should be layered very slowly and carefully to maximize interactions. So that is a very important point when performing Molish test. This is a very simple test, but the process will be very slow. The introduction of this H2SO4 will be slow, otherwise a fast reaction will be there. So chaining will be happening in case of this carbohydrate. So that will create a black ring. And even we perform the test correctly, and the alpha naphthol is concentrated more than what is needed, that will create a reddish violet ring. So carefully that solution is added slowly to minimize interactions, but to maximize the interaction with acid and the solution. Then Benedict's test. Composition of Benedict's reagent. Copper sulfate provides cupric ions. Sodium acetate provides alkaline medium. Sodium citrate provides precipitation of cupric ion. It's acting as a chelating agent. Principle: reducing sugars under alkaline conditions form indiols. These indiols are powerful reducing agents and are unstable. Hence, they decompose to yield a mixture of aldehyde that reduces cupric Cu2 plus ion to cuprous Cu3 plus ion as cuprous hydroxide CuOH. Principle: reducing sugars under alkaline conditions from indiols. These indiols are powerful reducing agents and are unstable. Hence, they decompose to yield a mixture of aldehyde that reduces cupric Cu2 plus ion to cuprous Cu3 plus ion as cuprous hydroxide CuOH. The CuOH during the process of heating get converted to different colored cuprous oxide Cu2 precipitate, which indicate presence of reducing sugars. Okay, 2 CuOH twice gives Cu2O plus CH2O. H2O, not CH2, just H2O. H2O. Benedict's test is also used as semi-quantitative test because the color of the precipitate gives approximate percentage of sugar excreted in urine. This is the test for identification of reducing sugars such as glucose, fructose, maltose, and lactose. This test is more sensitive test compared to other reduction tests. And Barfoit's test. Composition of Barfoit's reagent. Copper acetate provides cupric ions. Acetic acid provides acid medium. This is the reducing test, but it takes place in an acidic medium, mild acidic medium. In mild acidic medium, reducing sugars undergo tautomerization to form indiols which reduce cupric ions to cuprous ions. Cupric hydroxide is formed during the process of heating. Cuprous hydroxide is converted into cuprous oxide, which gives red precipitate. So monosaccharides react fast, whereas reaction with disaccharides is slow. Therefore, the test is used to differentiate monosaccharides and disaccharides. Higher concentration of disaccharides, about 5%, tend to give a positive Barfoit's test. So prolonged boiling for 7 to 12 minutes may give positive Barfoit's test for disaccharides. Then Benedict's test after hydrolysis. We are performing Benedict's test after hydrolysis. Perform this test only for sucrose. The Glycosidic linkage is easily hydrolyzed by dilute acid at elevated temperature. The products formed, that is glucose and fructose, reduce the Benedict's reagent to 1 ml of sucrose solution, add 5 drops of concentrated HCl. Boil the solution for 2 minutes, cool and then neutralize. Once again, I will be reading. To 1 ml of sucrose solution, add 5 drops of concentrated HCl. Boil the solution for 2 minutes, cool and then neutralize. Then hydrolyze it with 5 drops with 40% NaOH. Check using a piece of red litmus paper. Perform Benedict's test with neutralized sodium solution. Red precipitate of cuprous oxide is formed. Then Sullivan's test. Earlier we just saw the picture which depicts lactosazone, hedgehog shaped, soluble in hot water, yellow in color, crystallizes on cooling. Hedgehog shaped lactosazone. And maltosazone, sunflower shaped, soluble in hot water, yellow in color, crystallizes on cooling. Remember, like left, on your left side we have a hedgehog. So lactosazone, hedgehog shaped. Maltosazone, sunflower shaped. And remember, so fun, like marigold and sunflower have flowers. So Mary for maltosazone. Okay. So maltosazone, sunflower shaped, soluble in hot water, yellow in color, crystallizes on cooling. Then Sullivan's test, composition of Sullivan's reagent. Resorcinol with dilute HCl, principal HCl and Sullivan's reagent dehydrates the ketose to form furfural derivatives, which condense with resorcinol to form cherry red colored complex. This test is used to distinguish between aldoses and ketosis. Prolonged boiling may also give a positive Sullivan's test for aldose due to their conversion to keto group by HCl. And ozone test principle. When the reducing sugars are treated with phenylhydrazine, first phenylhydrazones soluble are formed. On heating, these hydrazones further react with 2 molecules of phenylhydrazine to form sugar ozones insoluble. Non-reducing sugars like sucrose does not form an ozone. For distinguishing the different reducing sugars in urine under conditions of glucosuria or lactosuria, this is the only test to differentiate between maltose and lactose. Only reducing sugars which have a phenaldehyde or keto group will form ozone crystals. Glucose, fructose and mannose form identical ozones. This is because these groups differ only at numbers 1 and 2 and carbon numbers, and ozone formation involves C1 and C2. So the difference between these sugars at C1 and C2 are masked during the ozone formation. Then glucosazone and fructosazone crystals are formed during boiling itself. So maltosazone and lactosazone crystals are not formed during the process of heating or boiling because ozones of these sugars are soluble in hot solution. Hence, ozones of maltose and lactose are formed only on cooling. Reactions of sucrose, we could see solubility test. First test, tested for solubility of given substance in water, soluble, absence of polysaccharide. And Molish test, take 1 ml of test solution in a clean or dry test tube and add 1 to 2 drops of Molish reagent, mix well and add 1 to 2 drops of concentrated H2SO4 along the sides of test tube without shaking. A reddish violet ring at the junction of 2 liquids, presence of carbohydrates. Then this carbohydrate is dehydrated by concentrated H2SO4 to form hydroxymethyl furfural, which condenses with alpha naphthol to form violet colored ring at the junction of 2 liquids. Hydroxymethyl furfural, chromogen is formed. Then Benedict test. Take 5 ml of Benedict's reagent in a clean dry test tube and add 8 drops of test solution and boil for 2 minutes. No colored precipitate. Absence of reducing sugar. Sucrose is not a reducing sugar. Then we are performing acid hydrolysis. Take 5 ml of test solution in a clean and dry test tube and add 4 drops of concentrated HCl and keep them boiling water both for 10 minutes. Cool under running water and then neutralize by adding 5 drops of 10% Na2CO3 till the effervescence ceases. Sucrose is hydrolyzed. Then Benedict's test after acid hydrolysis. Take 5 ml of Benedict's reagent in test tube and add 8 drops of hydrolysate to test solution. Mix and boil for 2 minutes. Brick red precipitate hydrolysate contains reducing sugars. Then Sylvanos test. Take 3 ml of Sylvanos reagent in a clean dry test tube and add 1 ml of hydrolysate solution of test solution and boil for 50 seconds. Cool the test tube. Cherry red color hydrolysate consists of keto sugars.